Hello, everyone, and thank you for joining our webinar on the Whole Mouse Brain Atlas publication package highlights. The program is shown on the screen. Please use the Q&A function for any questions. We'll save Q&A for the end and may not be able to get to each question. If you do not see captions, you may turn them on from the bottom of your Zoom screen. A brief survey will follow the webinar, and we appreciate your participation in that. Thanks again for joining our webinar. I'll pass it along to Hongkui Zeng to give the introduction.
Okay, hi everyone. Let me start sharing screen first. Oops. Okay, hi, good morning everyone. Welcome to this webinar on the BICCN Whole Mouse Brain Atlas publication package highlights. I'll give a brief introduction first. The BRAIN initiative set ambitious goal of understanding the brain by developing neurotechnologies to accelerate discovery. And the BRAIN initiative has set 7 priorities for its program. And the #1 priority of the 7 is to discover the diversity in the brain, meaning that especially discovering the cellular diversity across the brain. And the BRAIN initiative cell census effort following this priority was started in 2014 with its initial phase, BRAIN Initiative Cell Census Consortium, or BICCC, with a dozen or so pilot projects. And through the technology exploration pilot projects, we were able to set the stage for the generation of comprehensive brain cell type atlases. And that led to the second phase of the BRAIN initiative, the BRAIN Initiative Cell Census Network, or BICCN, which was running from 2017 to 2022. And the main goal of the BICCN was to use a number of single cell technologies from genomics to anatomy to connectivity to arrive at a comprehensive cell type atlas for the first mammalian species, the mouse, and also to begin pilot studies in the brains of human and non-human primate. And now, as we have completed the BICCN phase, we have moved on into the BRAIN Initiative Cell Atlas Network phase, which is also called BICAN or BICAN. And the main goal of BICAN is to complete the comprehensive cell type atlases for the larger primate brains, from human to non-human primate. So today we'll tell you about our work that has been done in BICCN, which is to complete this capstone project in generating a high resolution and multi-model cell type atlas for the entire mouse brain. We're very glad to say that we have completed this goal, and as published in 10 articles in Nature in December 2023, just about a month ago. This collective effort of over 200 investigators and scientists in BICCN has created the first complete cell census and atlas of a mammalian brain, and that is the laboratory mouse. So today you'll hear about a series of talks showcasing what we have found through this kind of comprehensive studies in those 10 papers. And here is just showing an overview of this publication package in Nature with several essays, introductory essays, there's a feature, editorial, as well as a news and views piece to talk about the significance of this type of work. And then following that, I'm just going to run a credit roll style introduction of the 10 articles in this publication package. And you'll see the 10 studies in the order it was presented in the journal. showcasing a variety of different kinds of molecular, anatomical, and also functional studies of the different cell types in the brain. So with that, I will turn over the podium to our speakers. Thank you.
Thank you for that great introduction. I'd like to introduce Zizhen Yao. Zizhen is an associate investigator in the Allen Institute for Brain Science, where she leads the computational efforts in categorization of cell type diversities in mammalian brains using single-cell genomics technologies. Before joining Allen, she did her postdoc at the Fred Hutchinson Cancer Research Center and received her PhD in computer science from the University of Washington in Seattle. Thank you. Take it away. And we're seeing your presenter view right now.
Okay, great. Yeah. And thank you, everyone, for tuning in.
Apologies. We see your notes. Sorry.
Okay, presenter view.
Perfect. Thank you.
Okay. Yeah. Thank you, everyone, for tuning in. It's a great pleasure to have this opportunity to present our work on building the mouse whole brain cell type atlas. So I would like to start with a high-level description of the experimental design. And this atlas is constructed by two primary data modalities, single-cell RNA-seq and spatial transcriptome. So to collect the single-cell RNA-seq data, we dissect the specific regions from the brain tissues and isolate the single cells for RNA-seq profiling and perform clustering to identify the cell types. To collect spatial transcriptomic data, we section the entire brain into the coronal sections and process each section using the technology known as Murfish. So this technology enables profiling expressions of hundreds of cells or hundreds of genes or thousands of genes at the single cell level while maintaining its spatial coordinates within the brain section. So through integration of these two data sets, we can infer the spatial distribution for every transcriptome cell type. So to make the atlas easier to navigate, we establish a 5-level hierarchy, starting the neighborhood at top, followed by class, a subclass, supertype, and cluster. Where is the laser? So our single-cell RNA-seq data sets include 7 million cells before QC and 4 million cells after QC. The median gene detection for neurons is more than 6,000 for 10x version 2 data set. And these high resolution, high data quality data sets give us the power to resolve in cell types extremely high resolution. So we have identified 5,322 clusters organized into 7 neighborhoods shown at the top of the tree, and then 34 class in the middle layer, and 305 subclass shown at the leaves of the tree. So regional composition for each subclass indicates that most of the neuronal subclass are dominated by one main brain regions. So here is another overview of the atlas visualized by UMAP, colored by class, brain regions, neurotransmitter type, and subclass. So cells for the same brain regions, as you see from the second UMAP, tend to cluster very tightly together, and the gradient in transcriptome space largely correspond to the spatial gradient. And this arrow recapitulates the actual anatomical structure of the brain, going from anterior to posterior to anterior along this axis. So orthogonal to the spatial gradient, you can also see the map can be divided into 2 halves based on the neurotransmitter type, with glutamatergic cells in one half and GABAergic cells in the other half. So the overall cell level landscape is largely shaped by the interplay of these 2 axes. So to build cell type hierarchy, our general strategy is to cluster the clusters at different resolution. A crucial decision in this process was determining which gene set to use. So we explore various options, including using transcription factor markers, functional genes such as neuropeptides and receptors, ion channel, etc., adhesion molecules, and all markers. We noticed that correlation based on transcription factors demonstrates superior modularity compared to other genes. This approach highlights greater contrast between closely related and distant cell types. Furthermore, the cell type hierarchy guided by transcription factors effectively recapitulate evolution, developmental origin, and spatial distribution. This outcome is perhaps not surprising, given that many transcription factors play key roles in cell type specification and spatial patterning during development. So to gain deeper understanding of logic for the cell type specification, it'd be very informative to identify transcription factor codes for each cell types. So before doing it, we first assess the predictive powers of transcription factors in distinguishing cell types. So on the left, we present 5-fold cross-validation accuracy at both the cluster and subclass level using different gene sets as features. Transcription factor markers, again, show excellent performance, suggesting that such code do exist for majority of cell types. To understand how transcription factors shaped the overall cell type landscape, we group them into gene modules. So this overview plot highlights several notable transcription gene modules specific to broad class of cell types, many of which are known to be the master regulators of those types. We have also identified transcription factors that are widely expressed, such as NR2F2, which delineates neighboring subclass or clusters, displaying local spatial gradient in many different parts of the brain. So for each subclass, subtype, and cluster, we present the transcription factor combo in the cell type annotation table, which can be useful to understand the regulation of these types. So as we explore this data set, we noticed neurons have different characteristics organized by different principles at different parts of the brain. So here are the UMAP for neurons in each neighborhood alongside corresponding Murphy sections and colored by subclass. So glutamatergic neurons in Palian show signature cortical layer structures, while the neurons in hypothalamus are organized mainly by the nucleus structure. So when comparing different characteristics of brain regions, we observed a strong dichotomy between dorsal and ventral parts of the brain. So the ventral part include hypothalamus, midbrain, and hindbrain, and the dorsal part include the isocortex, and thalamus, and cerebellum. So the ventral regions contain more but smaller clusters. The clusters are more similar to each other, measured by a smaller number of differential expressed genes, and they're spatially more confined. The dorsal regions contain fewer but larger clusters, show greater transcriptomic divergence, and spatially more widely distributed. So this distinct characteristic between dorsal and ventral regions likely suggests different evolutionary histories. The ventral regions are evolutionarily ancient and critical for survival, while the dorsal regions are more involved in adaptation. So we don't know the exact implications for these distinctions yet, but it'd be extremely interesting to validate regional differences across species and in development. Our atlas also provides a comprehensive catalog of all multitude neuronal types, and we observed very complicated co-expression pattern of these multitude of neurotransmitters with GABA or GLUTA or both. And here I highlight one subclass and the dopaminergic subclass, which shows all combinations. And cluster with different co-expression patterns have also distinct spatial distribution. So those were interested in this topic and find more details in the manuscript. While most of the discussion focused on neurons, this atlas also includes a high rest of the categorization of non-neural cell types. Many of these non-neural cell types also show remarkable spatial specificity. For example, almost all astrocyte types have distinct spatial distribution, and their transcriptome gradient reflecting the spatial gradient very precisely. The spatial localization of appendable cells are perhaps even more impressive, highlighting the cell type diversity in the ventricle areas of the brain that are previously underappreciated. So one current thing you have heard throughout this talk is really the high correspondence between transcriptomic identity and spatial localization. And here's a summary plot shows the subclass composition for each anatomical structures. And the correspondence is very clear. So we went further in the manuscript testing to what extent we can predict cell type identities based on spatial location and vice versa. And the results are even more striking. So transcriptome and spatial are really tightly coupled with each other. So there's a lot of information obtained from the study that cannot be described sufficiently in the manuscript and talks. So to make our data knowledge accessible to the whole community, we now offer various means for users to interact with this atlas with the help of the Allen technology team. The ABC atlas enables to visualize cell types in transcriptome space alongside with the Murphy sections. The GitHub page provides instructions for users how to access the data for data analysis, and MapMyCell allows users to up their own data to map the cells to reference atlas to gain the cell type annotation. So our work only marked the beginning of a bigger efforts to characterize cell types. Through support of BICAD, Alan is now extending our work to human and non-human primates to establish cross-species whole brain cell type atlas. We're also extending the temporal access to study development and aging and try to understand behavior and disease better in the context of cell types. Besides transcriptome, we have ramped up our data collections in other data modalities, including epigenetics, EFs and morphology, and connectomics. We've also started to accumulate extensive catalog of genetic tools for targeting specific cell types for functional assay. So this is, of course, a very ambitious goal, which requires a lot of extensive collaboration across the whole community. And by working with all of you, we are very excited to take on this challenge of building multimodal, spatial, temporal, and cross-species whole brain cell type atlas. And with that, I'd like to thank all the people who have contributed to this project, which is a lot. I'd especially thank Hongkui Zeng for her leadership, my colleagues from transcriptomic team, spatial transcriptomic team, program management team, and technology team. Also like to thank our collaborators from Xiaowei Zhuang's lab from Harvard, our funding agency, NIH. And that's all for my talk, and thank you for your attention.
Thank you so much, Zizhen. Next up, we'll have Xingjie Pan. Xingjie is a Jane Coffin's Child's Memorial Fund postdoctoral fellow and Dr. Xiaowei Zhang's lab at Harvard University. He received his BS in physics from Peking University and PhD in bioengineering from UCSF, where he studied computational protein design in Dr. Tanya Kortemi's lab. His current research focuses on developing deep learning models and high throughput experimental assays to predict and mechanistically understand how cell-cell signaling pathways affect cell fate determination during development. Please take it away.
Thanks for the kind introduction. Hi, everyone. Today, I will talk about our work on generating a molecularly defined and spatially resolved cell atlas of the whole mouse brain. We all know that brains of human and other mammals are highly complex. In order to understand such complex biological machines, we have to know what cell types does the brain have and how they are organized in the 3D space. So the major goal of our project is to generate a spatial atlas of molecularly defined cell types of the whole mouse brain. Using this atlas, we systematically characterize the molecular and cellular spatial organizations across the brain. Also, we systematically profiled cell-cell interactions and communications in each major brain region. So in order to generate the whole mouse brain atlas, we first select a subset of genes that can distinguish all transcriptionally defined cell types in the mouse brain. We selected 1,100 genes with the help from our collaborators from the Allen Brain Institute, using their single-cell sequencing data. Next, we performed the Murphy experiment on 245 brain slices from 4 mice. For each brain slice, we imaged the mRNA molecules of the 1,100 genes in each cell. Next, we integrated the 4 million cell RNA sequencing cells and the 9 million Murphy cells. The integration basically co-embeds the single-cell sequencing data and the Murphy data in the same space. Then we can transfer the cell type labels from the sequencing cells to the neighbor Murphy cells in the co-embedding space. And using the neighbor sequencing cells in this embedding space, we also imputed the transcriptome-wide gene expression for each Murphy measured cells. Finally, we registered the cells to the Allen Brain Common Coordinate Framework, such that each of our cells gets a standard coordinate. By doing so, we build a spatially resolved single-cell atlas of the whole mouse brain. So we have identified cells that belong to about 5,000 transcriptionally defined cell clusters, which can be further grouped into about 300 major cell types. We call them subclasses. For each cell, we have a transcriptome-wide gene expression profile, and we can identify the cell type and their marker genes, such as neurotransmitters. And we have the XYZ coordinate in the 3D common coordinate framework space, from which we can derive their brain region identities. We can also look at the cell's neighbors to find cell-cell interactions. So using this atlas, we systematically characterize the molecular and cellular spatial organizations across the whole brain. So our atlas reviewed highly diverse spatial organizations of neurons, both at the 300 subclass level and at the 5,000 cluster level. So basically, different transcriptionally defined neurons often have distinct spatial distributions. So we comprehensively determined the spatial dependency of neurotransmitter usage. So glutamatergic and GABAergic neurons together make up about 98% of the neuronal populations. And both glutamatergic and GABAergic neurons were widely distributed across the whole brain and can be classified into diverse cell types with distinct spatial distributions. And we also find that neurons expressing modulatory neurotransmitters account for about 2% of the total neuronal population, and they also showed strong regional specificity. Besides the neurons, more than half of the brain cells are actually non-neuronal cells. We identified 117 clusters of non-neuronal cells belonging to 23 subclasses. Some of the non-neuronal cells also showed strong regional specificity. One of the example is the astrocytes. And here in this slide, I'm showing you that almost all the astrocytes clusters have very distinct spatial distributions. And after observing that different cell types have diverse spatial distributions, we wondered such a question: if we use the transcriptionally defined cell types to define brain regions, how well they compare to the canonically defined brain regions by anatomy or functions. To this end, we defined spatial modules by clustering the cells by their by the cell type compositions in their spatial neighbors. So at the coarse grain level, the spatial modules are basically the major brain regions. But one notable discrepancy is between the midbrain and hindbrain. This discrepancy originated from the gradual changes of cell type compositions from the midbrain to the hindbrain. So this makes the ambiguous determination of the midbrain-hindbrain boundary very challenging. And at the finer level, the spatial modules largely agree with the regions defined by the common coordinate framework, but there are also some discrepancies. Those discrepancies, on the one hand, could due to our higher molecular resolution from our atlas, and on the other hand, some CCF regions, such as many isocortical regions, are defined by their connectivity and functions. These regions may not be transcriptionally distinct. So when we do cell typing by clustering or defining spatial regions by spatial module analysis, we kind of tend to classify cells into discrete states. But in many cases, cells form a continuum in both transcriptional and spatial dimensions. We identified spatial gradients in many brain regions, for example, in the isocortex, in striatum, and even in ventricles for both like neuronal cells and non-neuronal cells. So finally, we use our atlas to systematically profile cell-cell interactions and communications. We focus on the short-range cell-cell interactions. So here we consider a pair of cell subclasses that are connecting to each other if their soma contact frequency is significantly higher than random chance. We determined the random chance by performing a local spatial coordinate randomization. So for each pair of contacting subclasses, we also identified the ligand receptor pairs that are significantly upregulated in the contacting cells versus the non-contacting cells. And we found hundreds of contacting pairs of subclasses, both for neural, non-neuronal interaction, non-neuronal interaction, and neuron interactions. And here I'm showing you some examples for each of these categories. So basically, we find significant interactions between endothelial cells and BAM, which is a type of macrophages. And we also find interactions between astrocytes and immature neurons in the olfactory bulb. And we found neuron significant contacts in the midbrain. So here I'm showing you a specific example between endothelial cells and macrophages. We identified overexpressed ligand receptor pairs belonging to a few signaling pathways. One of the top pathway is the laminin pathway. Because basal laminin are known to promote macrophage maturation, this result could indicate that endothelial cells actually uses laminin to maintain the brain macrophage pool. We also find that a gene like CYTL1 that encode a cytokine is upregulated when endothelial cells are contacting with macrophages. This observation also indicates that endothelial cells might use this cytokine to recruit macrophages in the brain. So we found we also found that different vascular cells actually preferentially interacting with different immune cells. Here I'm showing you two types of vascular cells, the endothelial cells and pericytes, as well as two immune cells, the macrophages and microglia. So although both vascular cell types interact with both immune cell types, the probability of contacts are different. The pericytes have a higher probability to interact with microglia, while the endothelial cells have a higher probability to interact with macrophages. So we think our atlas will be a valuable resource for the community. So we have made our data publicly available, both from the Allen Brain Cell Atlas and Cell by Gene. And in summary, we have generated a spatial atlas of molecularly defined and spatially resolved atlas. And the atlas allowed us to systematically characterize the molecular diversity and cellular diversity across the entire mouse brain. And we have identified hundreds of cell type specific cell-cell interactions. So in the end, I want to thank members from the Zhuang lab, my PI Xiaowei Zhuang, and my collaborators Meng Zhang, Wang Zheng, and Aaron Halpern, who contributed significantly to this project. I also want to thank our collaborators from the Allen Brain Institute, especially Hongkui Zeng and Zizhen Yao. And I want to thank our funding agency. Thank you for your attention.
Thank you so much for that talk. Next up, we have Jonah Langlieb. Jonah is a computational associate in the McCosco lab at the Broad Institute, where he led the co-analysis of this mouse atlas with Nina Sajdev. Before joining the Broad, he received a BA from Swarthmore College. Please take it away.
Thanks for the kind introduction. Hi, I'm Jonah. So today I'll be talking about our recent paper, The Molecular Cytoarchitecture of the Adult Mouse Brain, and the construction and analysis of this reference data set, which is a brain-wide cell type atlas of the mouse brain. It represents a 5-year-long effort in new technology development, large-scale data generation, and computational analysis. I'm going to start with acknowledgements. This is only made possible by so many amazing members of the lab, of the McCosco lab, as well as members of the Fei-Chen lab and our collaborators in the Park Features lab. I think of my role really just showing off how cool the data they generated is. So there are two foundational technologies that the McCosco lab helped develop, which we used in this analysis. The first technology is one that many of us know and love, and it's droplet-based single nuclei RNA sequencing. And so I won't go into much detail here, but at a high level, this gives us an unbiased view of the mRNA molecules in isolated nuclei. At a lower level, this involves cool technology like microfluidics, which I mentioned mostly as an excuse to show this fun animation of a cell going on its microfluidic roller coaster ride. The second technology I'll talk about in some detail is SlideSeq. So this is developed with Fei-Chen's lab and is our high throughput, unbiased spatial transcriptomic assay. This lets us create a spatial map of the gene expression of the cells in different tissue slices. So to move on to the specific data sets in this paper, we knew that creating this large-scale data was theoretically feasible, but it was still a truly heroic effort to get it done. And so over a couple of years, led by Chuck Vanderberg and Naim Adolf, the lab dissected the mouse brains into 100 distinct sections called dissectates. And in the end, we generated 6 million single nuclei RNA-seq profiles from these dissectates. And on the right is a downsampled t-SNE of these nuclear profiles, colored by their major cell class. Previous presentations have shown such plots, but as a reminder, each dot here is one cell, and they're put into this 2D visualization, organized by how similar a given cell's mRNA expression is to the other cells in the dataset. And we can see that nearly all the molecular diversity in the brain was neuronal, as previously well documented, shown by the abundance of pink dots in that brain-wide t-SNE. While I'm here, I can also quantify that. And we see we have around 4,800 unique neuronal cell types, coming to 2.5 million neuronal cells. As for some reasons why we hope this single nuclei type of data will be valuable to the community. For one, it aids in the compatibility with future human data, such as with the oncoming BICAN grant. We also found that its association procedure was consistent across tissues, which further helps be consistent in the proportional representation of individual cell types. This is especially valuable when looking at very rare neuronal cell types, and there are in fact a lot of such small clusters in our data set. To switch gears in our whirlwind tour, I'll talk about the SlideSeq data. And as I said, this was led by Carol Badarama and Evan Murray in collaboration with Fei Chen's lab. And they took a single female mouse brain and carefully and precisely cut it into 101 serial sections, going from the back to the front of the brain. They also included separate pre- and post-microscopy images, missile stains, which sandwiched each of these SlideSeq slices. And then Mukund Raj, in collaboration with Daniel Tward in the Partha lab, aligned these missile stains and SlideSeq slices into one cohesive 3D space, both within themselves and to the Allen Institute's very nice CCF framework. To focus on a second on the nitty-gritty aspects of this analysis, of this assay, it gives 10 micron or roughly cellular resolution spatial pixels. It gives untargeted or unbiased whole transcriptome output of giving a digital gene expression output. It's also high throughput, and you can think of it a little bit like a single new cell with some batch effects with spatial barcodes added. And if you want to see this idea of single cell with the addition of spatial barcodes taken to its next logical step, we also have a new slide tags assay in collaboration again with Fei Chen's lab, which you can think of as exactly that. As one note, a lot of our paper and the rest of this talk will focus on cell type mappings and the SlideSeq data, which all comes from the foundation of these individualized gene localizations, which we found very helpful and interesting in their own right. And so on the right, I show one single gene, TPH2, which is especially useful when combined with, say, the Allen Institute's ISH data. To then quickly talk about the cell type integration, we wanted to map these thousands of single nuclei clusters onto all the SlideSeq spatial pixels. And so we adapted an algorithm called RCDD, which is developed by Dylan Cable in the Fei Chen lab. To quickly give you a sense of the quality and accuracy of these mappings, here are the predicted spatial distributions of 47 different cortical excitatory cell types, which are this very stereotyped layer distribution, which is previously well studied. We've ordered them here by their positions based on this prior integrative analysis. And you can see that these newly mapped layer depths very strongly match these expected order, giving us some confidence that the mapping is reliable and robust. And so now we're in a position where we can look across thousands of cell types we identified in our single nuclei atlas and see how they were spatially distributed. And so I can dive more deeply into individual cell type localizations. Here showing some of these highly specific mappings. So on the left here are the cell types spatially mapping onto individual SlideSeq slices. And on the right is the same cell types highlighted in the global 2D t-SNE visualization from before. We added an inset to better see these intricate cell types hidden in this global t-SNE. So these plots show cell types from a variety of regions, from the thalamus on the upper left, medial raphe nucleus on the upper right, and the piriform cortex and cerebellum on the bottom. We can also visualize this globally in a couple different ways. And I'll walk you through some of them. On the left is a heat map, where each row is one cell type, and each column is one fine-grained region, where the higher level regions are given by the color bar at the top. We color this heat map by the fraction of the mappings for each of those cell types in each region. And as a reminder, what we're summarizing, each of these small matrix squares is actually a collection of the heat maps shown on the previous slide on the bottom right here. And overall, we found there's a lot of focality in these map cell types, especially below the cortex, in that most cell types or most rows are only in a few regions or a few columns. Here's another visualization of showing a similar aspect. We can, it's the same 2D plot as before, but now individual cell types are colored by their main brain regions they map most to, aggregating again upward from the underlying most detailed SlideSeq localizations to these top structures. And you can see on the top here are these green telencephalic populations of the cortex, hippocampus, and striatum. And on the bottom, there's absolute morass of cell types in the hypothalamus, midbrain, and hindbrain. We interpret this more mathematically in the paper, but even visually, you can see how intricate these midbrain and hindbrain clusters become. I can say personally that when I look at this delicate intricacy of these plots and all the complexity of the midbrain and hindbrain shown here, it really hits home what an interesting challenge it will be in the future to understand this in a greater detail. But that's the name of the game. And so we're excited to work on this challenge with all of you and see how we can work together to understand this amazing complexity shown in this and the other papers in this collection. As one final visualization, without belaboring the point too much, because I think it's of interest to the group and it can be useful in other companion analyses, we also aggregated the cell types hierarchically into a tree. and color each of these low-level leaf nodes by the top region. You can again see this massive, incredible amount of midbrain and hindbrain cell types, that pinkish and red color, all forming this incredibly detailed cell type hierarchy. We can also see that the tree is broadly segmented into main regions as expected. So now we have a pretty complete picture of where these cell types are located throughout the brain. And so we wanted to know what principles we can extract of brain biology from these transcriptional profiles. This is spearheaded by Nina Sachdev, who's off to grad school. And she did lots of these interesting analyses, such as looking at the principles and nature of neurotransmission across these different areas of the brain. We can also look at these single nuclei profiles and learn about the activity-regulated gene expression across different cell types, and bring in broader data sets, such as when we did a systematic analysis of the enrichment for gene loss-associated genes across brain cell types. We can even use this comprehensive nature of the atlas to identify optimally small covering sets, which could be useful in future assays to identify and probe these cell types. To look to the future, I wanted to illustrate the magnitude of this incredibly rewarding challenge we face. And so here's a little anecdote about just one of these thousands of new cell types we describe in this data set, in this case in the cerebellum. A prominent cell type in the cerebellum are molecular layer interneurons. In our previous work, we had found clear evidence of two distinct cell types in the molecular layer, which we called MLI1 and MLI2. And in that original work, we describing this new cell type, we show that MLI2s have several distinct physiological properties. But of course, our collaborator, Wei Roger, was eager to understand what distinct role these cells had. It took 3 years of physiology, mouse transgenesis, EM reconstruction, and mouse behavior work to learn that MLI2s actually represent an entirely new circuit feature, which was previously unknown in the cerebellum, which is inhibition, and could potentially provide a circular explanation for learning and plasticity in the cerebellar cortex. And so this is all very cool, but it's not necessarily going to scale to the kind of workups we will need to understand the thousands of new cell types we describe in this and the other papers, and how they function more generally. And so we've been working hard developing methods that would allow us to start annotating these new cell types with greater ease and greater throughput. And we're excited to see how these can work together with the rest of the community to understand, to get a handle and understand this beautiful complexity of the mouse and mammalian brain. As a final note, we worked hard to make a custom portal for our data set at braincelldata.org, which we hope will allow practitioners to seamlessly look through the spatial distribution of cell types and genes, as well as to quickly narrow down on the cell types of interest, given either a region or a marker gene. And so again, this is available at www.braincelldata.org, and we hope it was useful, and we'd be interested in any feedback you can give about how you find using it. Thanks. Again, this is only possible because of the amazing, incredible work by so many different lab members and the collaborators. In particular, I want to call out Nina Sachdev, Mukund Raj, Chuck Vanderberg, Naima Daf, Carol Valderrama, and Evan Murray. Thank you.
Thank you so much for that talk. Next up, we will have, next up, we will have Hanqing Liu. Hanqing is currently a Harvard Junior Fellow. Before joining Harvard, he was a postdoctoral fellow in the Ecker Lab at the Salk Institute for Biological Studies, where he's got his PhD in biology from UCSD under the guidance of Dr. Joseph Ecker. His doctoral research focuses on developing single-cell multi-omic technologies and analytical frameworks to investigate and decipher the epigenomic diversity of mammalian brains across spatial and temporal dimensions. Please take it away.
Thank you for the introduction, and it's my great pleasure to have the chance to present here. I'm going to go through our work performed in the Ecker Lab at Salk Institute, which is aiming to build a DNA methylation and a 3D multi-omic atlas for the entire mouse brain. So as I mentioned in this talk, previously you have heard a few transcriptomics, single cell or spatial data set. And in our particular study, we add additional modalities from the epigenetics. One is the DNA methylation. In particular, when we talk about methylation in brain, we separate the CG and the CH. The CH methylation is a unique modality, especially in the neuronal genome. And on the other hand, we are also adding the chromatin conformation information because through our brain atlas profiling, we observed a lot of cell type specific chromatin structures at multiple genomic resolutions. So with that goal, in the BICCN project, we used the Y-type adult mouse brain and performed the brain dissection to slice it into 600 microns and get anatomical regions. we then separate nuclei to perform either the single nucleus methylation technology or to perform in situ chromatin conformation capture to allow us capture methylation and 3D genome information within the same cell. We then sort the cell into 384 well plates by neuron positive enrichment. So we get majority of the data from the neurons, but we also cover sufficient number of non-neurons in each of the region. And then we perform bisulfite conversion, which is a critical part, chemical step to turn unmethylated cytosine into uracil and methylated cytosine as unchanged so that we can read out the DNA methylation status in base resolution. And then undergo our library prep through automation and sequencing. And our dissection framework of this project is, as I mentioned, to slice the brain into 600 micron slice and take anatomical regions. In total, we have 117 brain regions, which covers all the major anatomical structures in the entire mouse brain. And we, after the completion of this project, we get a total of more than 300,000 single cell methylation profiles, and also 176,000 single cell methylation and chromatin conformation multi-omic profiles. So we begin with analyzing the data by doing the cell iterative clustering using the DNA methylation, as it is a shared modality in both technologies. And this, after the clustering and the integration, these two data sets agree with each other very well, as indicated by this t-SNE. And we performed iterative clustering using the cell by 100 kb bin features, and both as MCH and MCG info modality to identify cell clusters at multiple resolution. And after annotation, together with the single cell RNA data and the cell taxonomy provided by our collaborators in the Allen Institute, we can get a total of more than 200 cell subclasses and more than 4,000 cell groups. And in order to understand and annotate these data sets in a large scale, we performed cross-modality integrations using gene body information to integrate with single-cell RNA data set that just presented by Zizhen in the previous presentation. And we also integrated with single-cell attack data, which is generated in BICCM from Bing Ren Lab, as you will heard in the next presentation. And through this integration, which allow us to cross-validate our cell clustering and also match different modalities together for each of the cell group or cell subclass that we identified. And here is an example of the integration UMAP as plotted by using all the neurons from each of the data set. And in the top row is our methylation or 3D multi-omic data set. And in the bottom row is the single cell attack and the single cell RNA data from the Allen Institute. And these cells are colored by the same cell subclass taxonomy so that they share the same palette. And that allows you to visually understand that these data are very well matched to each other. And to show you the same integration result in a confusion map, on the right, this is a aligned result from where each of the columns or the x-axis are the thousands of methylation cell groups, and the y-axis are all the transcriptome cell clusters at the highest resolution. And to zoom in to show a particular two particular cell subclass as example, you can see that in this layer 5ET cortical glutamatergic cell, which is the two plots here, the DNA methylation and RNA data can match with each other pretty well, and the clusters identified separately from each modality really have a pretty good correspondence, although it's not always one-to-one correspondency, but it does show that there's a lot of detailed cell taxonomies that can be matched by both modality. And another example for demonstrate the same idea, which is another cell from midbrain and medulla. So together, after the integration and annotation, we get we finished the generation of our single cell methylation and the multi-omic data set. And one additional information which you also heard from other talks is the spatial location of each of the cells. And here I'm showing you all the DNA methylation cell in their own t-SNE plot, but without color. And I will next show color them one by one using the anatomical metadata that we get through our manual dissection. And in this plot, in this way, you can see that the DNA methylation cell by itself really can be separated very well through their spatial locations. And these information, which are created through our manual dissection, provides a very good ground truth for us to understand that the data and the cell clusters is very spatial specific. And later, to further investigate the correspondency between the DNA methylation profile and the cell's spatial location, we also performed the Murphy experiment by designing a gene panel that is variable that is spatially variable identified from DNA, their gene body methylation level, and then perform the integration between the single cell methylation and their corresponding Murphy experiment and the cells from the corresponding Murphy experiment. And then we can impute the spatial location of the DNA methylation cell after this integration analysis. And to show you six coronal slides as examples, here in the first row, I'm plotting the 3D anatomical structures colored by our manual dissection, which means when we do the single cell methylation analysis, we are manually taking slice at this position and cut the slice to get our sample in the colored diagram. And in the bottom two rows, I'm showing you the single cell methylation data imputed and mapped to the spatial coordinates of the Murphy data, and then colored by their spatial dissection label from the experimental metadata. And the glutamatergic cell and the GABAergic cell are plotted separately on these two rows. And this result, together with the metadata labeling, really confirmed that our DNA methylation cell has a lot of spatial information, which allow us to spatially map them to the Murphy data in a pretty precise level. As you can tell, that the cortical excitatory cells can display a certain level of arealization as we dissect it. And in the subcortical areas, the GABAergic cell can also be mapped into pretty precise brain nuclei in, for example, the thalamus and hypothalamus. And so far, I'm showing you after generating the epigenetic data set, we observed a huge amount of cellular heterogeneity and spatial specificity. But in addition to that, our data set is also a genome-wide epigenetic atlas. And this allows us, when we have hundreds of thousands of cell types, with each of them have a pretty good genome profiling, we can use the cell type as cell types as features to understand more genomic regulatory regions in a high resolution, which is not possible before without this data set. And to showcase the power of this matched multimodality data set, I'm using one particular gene, TLE4, as an example. And this gene is a marker gene of this circled cell type here, which is layer 6 T cell, and another circled out negative cell population as a comparison. And on the left, you can see the gene body RNA expression level and gene body MCH level. And on the right is a nearby cis regulatory elements with the attack accessibility signal and the methylation signal of the MCG.
Just want to let you know you're at time, if you could wrap up your talk.
Yeah, okay. Yeah, and these modalities will match together. And in addition, we also have the 3D genome to link the regulatory elements together with the gene body. And sorry about because of the time, I have to skip. But with this matched modality, we can construct a gene regulatory network and predict the key regulatory elements for each of the cell type through multiple information. And certainly, if you're interested in more details, please go to our paper. And there is also a website for visualize the data set and more details about the data download. And with that, I want to thank everyone. It's a huge teamwork and all our collaborators in BICC and funding sources. Thank you.
Thank you so much for that talk. I'd like to introduce next Songpeng Zu. Songpeng is a postdoc at the Bing Ren Lab at UCSD. He got his bachelor's degree and PhD at Tsinghua University in China. Before joining Bing's lab, he had 3 years of industry experience in deep learning. During 2019 to 2021, he also worked as a postdoc in Xingjie Liu lab at Harvard. He focuses on the applications of statistical machine learning on gene regulatory programs and drug discovery. Please take it away. Thank you.
Hi, everyone. Thank you for the gentle introduction. I'm glad to be here to introduce our work about the chromatin accessibility analysis in the adult mouse brain. And this work together with Yang and Carla in our lab. And I want to, so this work actually, well, the huge amount of work from several teams. So basically we share the same samples with single cell methylation data set generated by the market team. And also the sequencing part is generated by our Center for Epigenomics. And we also get lots of help from Joe Acri and the Allen Institute for the comments and also the data sharing. And also lots of members in our lab joined this project and did lots of good work. So in our work, well, we mainly want to identify the cis-regulated element across the whole mouse brain. And the cis-regulated elements are the DNA elements that usually can be targeted by the transcriptomic factors. And they are kind of promoters and enhancers. They control the cell type specific gene expressions. And one of the key features is that typically the chromatin region in this part should be open. And so that we use the chromatin accessibility to measure this kind of cis-regulated elements or the potential cis-regulated elements. It turns out that not only they are important for the cell type functions with specific functions, they also can be the target. So based on the recent studies, like gene editings, so people can target the enhancers upstream of a transcription factor to reduce the expression of the corresponding transcription factors. So in our study, we use a mouse as a model to study the cis-regulated elements across the whole mouse brain. And here, I will show that. So with the same dissection collected by the market team, in total, we get 2.3 million cells across the whole mouse brain. And based on our integration, clustering, and the analysis using the SnapAttack2 method, we identified more than 1,400 cell subtypes. And the left part corresponding to UMAP in our data. So you can see that each dot is actually corresponding to one cell and colored by the corresponding major regions. And here's the corresponding non-neuron cells and the glutamatergic neuron cells and GABAergic neuron cells. On the right side, I also list the corresponding t-SNP plot from the Allen Institute to compare the different modalities. And after that, we do the annotations for our data using the integration with Allen's single-cell RNA data. Basically, we use the COH version 5 to do the integration, and we show this kind of co-embedding space with a major region, and also we see the beautiful consensus metric to see that for each cluster in our case, we have the corresponding annotations from Allen Institute with multiple hierarchical levels. And after that, we actually identified for each of the cell subtype, we do the conserved peak calling, identify the cis-regulated elements. And in total, we identified 1 million cis-regulated elements, which covering almost 19% of the mouse genome. And more than 40% of the cis-regulated elements haven't been reported by previous bulk level study. And here, so you can see this is the corresponding the heat map to show the peak modules, and each row corresponding to the cell subclasses, and each column corresponding to the peak or the candidate cis-directed element. And the color here corresponding to the chromatin accessibility. So based on this heat map, we show that the cis-directed element can have some kind of cell type specific patterns. And we can see that very similar pattern from the single cell methylation data. And after that, our aim is trying to identify the enhancers. We do this kind of identification based on the correlation study with our single cell ataxia data and the single cell RNA data from Allen Institute. You can see here, so the heat map for each row actually corresponding to the one potential enhancer, and each column to show their chromatin accessibility in different subclasses. And also on the red part is corresponding to the target gene, potential target gene, and their gene expression in the corresponding subclasses. You can see that they have very high correlation between each other. And we also validate part of this kind of connections based on the single cell nuclear M3C data to see they are indeed close to each other in the spatial data set. And in the meanwhile, we're also using CellOracle to identify the to identify the gene regulation programs in the cell type level. But due to the time, I will not show the data here. And one interesting part is because we are studying mouse, but mouse is actually a very good model to study human diseases. In our previous paper, which Yang did lots of work to do the comparison with human and mouse part. So based on the cerebral regions, you can see here, lots of the most human region and mouse region, they show the kind of conservation, and especially for the cis-directed elements. First of all, Yang also did the corresponding JIVA study to identify the cell type that are important for the potential neurological diseases. And interestingly, that lots of this kind of non-coding region related with the neurological disease are actually conserved in mouse, also in mouse genome. In our study, so we now have the whole mouse brain. We mapping our data cis-directed element to the human genome, and 58% of them show the orthologous, and especially about 22% of these cis-regulated elements can be mapped to the cis-regulated element in the human genome. That's pretty interesting. And last, we actually do the deep learning model to say if we can, using the raw DNA sequences to predict the corresponding chromatin accessibility in a cell type specific level. And this, if this works, it actually can be used for in silico perturbation study to study the gene editing part. And we do this part, so we basically using the Bayesian model and do the corresponding cell type specific learning and partition the genome into different bins and predict the corresponding chromatin accessibility. So this part show the corresponding signal. This is the true signal we have seen in different cell types. And this is the signal we have seen well predicted by our model. So you can see we can not only predict the global patterns, the global chromatin accessibility showing each of the cell types, we can also successfully predict the corresponding specific cell type specific chromatin accessibility signals. And furthermore, we extend our model. Actually, we didn't train our model in the human data set. We just directly using our model train on the mouse part and apply to the human data set. And you can see here, this is the true signal from the human data set with the corresponding cell type. And this is the predict signals using our model, which train only on the mouse part. And you can see we indeed can also capture the signals in human model. So this in human genome. So this is pretty interesting. Okay, so this is the last slide for my today's presentation. And our data are shared in the NCBN. Also, we have a catalyst to organize our data. And finally, we have already uploaded all the code and the corresponding links to our data in the GitHub repo. So feel free to submit any issues in our GitHub repo. And thank you so much for today's for your attention. That's it.
Thank you so much. Next, I'd like to introduce Jingtian Zhou, who's a science fellow at the Arc Institute. He received his BS from Tsinghua University and his PhD through the Bioinformatics and Systems Biology program at the University of California in San Diego. During his PhD, he worked with Dr. Joseph Ecker to explore the gene regulation landscape of the mammalian brain through integrative analysis of multimodality single-cell data. At ARC, he will continue to develop single-cell high-throughput technologies and computational tools in his own lab to come up with the more diverse types of data and analyses for quantitative modeling of gene expression dynamics. Please take it away. Thank you.
All right, thank you for the kind introduction. And today I will talk about our recent progress using a technology we call epigenomics. So to link the large-scale molecular-defined cell types with neuronal projections. So this is one of the work I did when I was in Joe Ecker's lab, together with Dr. Ed Calloway's lab. And so we know that traditionally neuron cell types could be defined anatomically by their projections and connections, which could be tightly associated with their physiological functions, demonstrated by research in the past decades. And we also heard in previous talks, neuronal cell types could be classified by their transcriptome and epigenome in high throughput way. But what is the relationship between these two definitions of cell types remains less characterized. So our goal is then to link the molecular cell types with their distal projection targets. The approach we took is epiretroseq. As you can read from the name, it combines the retrograde virus tracing with epigenome or DNA methylation sequencing. So we inject the retrograde virus in the target brain regions, and it has a crate that can activate the GFP expression in the nuclei in the source. So then we select the GFP positive neurons for methylation sequencing, and for each single neuron, we know both the projection targets and the epigenome. So we apply this method to 32 sources and 24 targets covering all major brain structures and sampled in total 225 projections. The direct sampling approach in previous talks told us the difference of cell types across brain regions, but with our data set with enriched projections, we can go a step further to ask a few more questions. At the cell level, we're asking whether the neurons projecting from the same source to different targets can also be distinguished from their epigenome. And reversely, when we group the cells into hundreds of subclasses and thousands of clusters, where are these cells primarily projecting to? At the genome level, basically we're asking what genes and regulatory elements are differentially activated in neuronal types projecting to different targets, and what transcription factors could be driving these differences. As a starting point, I will show you an example of a comparison of intratalancellic, the IT, and the extratalancellic, ET neurons. So the neurons projecting to basically cerebral targets versus the neurons projecting to brainstem. And in the cortex, we know that this is the most explicit distinction of the projection types in molecular space, with IT projection neurons clustered in this layer 2-3 to layer 6 IT clusters, while the ET projection neurons clustered in the SINE layer 5 ET cluster. But when looking across the whole brain, we started to see a distinct level of separation between IT versus ET neurons in different source regions, and they're not always in separate clusters. To quantify this epigenome difference between neurons projecting to different targets, we train a model that uses DNA methylation as features to distinguish the neurons projecting to two different targets, and quantify this model performance with AUROC as the distinguishability between the targets. So then we saw this IT and ET neurons are very distinct in some cortical sources as well as in the thalamus, while some other sources like pallidum and hypothalamus, they are not distinguishable as good, suggesting these projection differences are source dependent. And next, we ask whether the difference between IT versus ET are shared across sources. So to achieve that, we train this prediction model in one source and measure the performance in another source. So the results show that the source form these small groups that share the projection difference. For example, this isocortex and archicortex form their own groups, as well as olfactory area and different parts of the thalamus, suggesting they share this IT versus ET difference within the group. And we further look at the differentially methylated genes between IT versus ET and saw that the sources from the same group, like those cortical regions, often have similar differential pattern of these genes. Well, these genes could have a reversed direction in the source regions like thalamus. And next, we ask what genes are the dominant contributor to this predictability. We choose 5 gene categories and use each of these gene categories as features to compare their target prediction accuracy. So these different categories have different number of genes and use more genes tend to increase the prediction performance. So to control this bias, we downsample the larger gene categories into the same number of genes as in the smaller gene categories to facilitate the comparison. For example, here we downsample all the categories to only 19 genes to match the number of neuropeptide and receptors. And we observe that the neural projection development genes have the strongest target prediction power, then followed by neurotransmitter receptors, the ion channel, then neuropeptide receptors, transcription factors, and lastly, the random selected genes. And when using more genes, the ranking of the terms stays the same. So we think this aligns with the idea that the difference between projection neurons are tied to this developmental mechanism that's specifying projection targets. But interestingly, although we usually consider transcription factor as an important gene category that decide the cell types, as also shown in some of the previous talks, so this result could suggest that transcription factor may not be the driving factor for neural projections. And in the next part, to show how we can link the consensus cell clusters defined across BICCN with our the projection types, I will start with a simple example in midbrain VTA. Here is like a joint embedding of RNA and DNA methylation colored by the predicted expression level of the marker gene of glutamate, GABA, and dopamine neurons. So we map the epiretroseq data to this embedding. We see the relationship between projection neurons and these neurotransmitters, like a cluster emphasized here, the triple positive for these three neurotransmitters is not enriched for any projections, while the cortical projection neurons are enriched in the glutamate and dopamine double positive cluster. And this striatum projections are enriched in the dopamine-only cluster, and the ET cells are in these GABAergic clusters. And a more comprehensive example is in hypothalamus. Again, you see these epiretroseq cells are not in all the cluster, but only a subset of them. So we can further quantify the proportion of cells in each clusters for the different projection neurons, and we see like neurons having different projections are enriched in different clusters. For example, this cluster 76 on the top is enriched for neurons projecting to striatum and pons, while the cluster 3 is enriched for neurons projecting to thalamus. But further integrate this data with merfish, we see these clusters are located in the stripe structure in zona incerta at the posterior part of the hypothalamus. And we also see other projection enriched clusters located in the specific locations, especially on this sagittal slice, like a topographical distribution of these clusters enriched for different projections. And this data show how we can spatially locate the projection associated cell clusters with a much higher resolution than the dissection we are able to make. As a last piece, I feel it's also worth mentioning some of the negative results. Like Kim et al. Paper used the ACT-seq to associate the ventral medial hypothalamus, the VMH cell types with mouse behaviors. So we integrate the data with ours to see if any of this projection enriched cell types are associated with these behaviors. So Kim et al. Enriched for cells from only part of the VMH, and only reported some of the VMH VL clusters that are linked with false expression changes. But our data don't show any of these VMH VL clusters enriched for neurons projecting to the targets we profiled. And when examining all these VMH clusters, we only see 2 of the projection enriched clusters that are barely significant associated with the aggression and social fear. However, I think this does not rule out the possibility that we will need to zoom into higher resolution subtypes and injections to observe the full link between molecular projections and behavior. So due to the interest of time, I cannot go over all these use case and finding of our data set. If you're interested, we definitely encourage you to go to our paper and explore our data and browser. And as a final summary, I've shown you that epiretroseq revealed a source-dependent molecular difference between neurons projecting to different targets, and this integrative analysis enabled mapping spatial location and the projection target of the neuronal clusters. So this will serve as an important step in building this multi-model cell census of the whole mouse brain. Although we have shown many quantitative links between the projections and molecular cell types, to fully understand the molecular principle governing neuronal projections and linking them further with the functional data is still kind of challenging, which would need more specific study design and improve the technology and achievement. With that, I want to end by thank all this team that really make this huge effort come true. So that's it. Thank you.
Thank you so much for your presentation. I'd like to next introduce Carla Winter. Carla is an MD PhD candidate at Harvard Medical School. She completed her PhD in the laboratory of Zhigong He at Boston Children's Hospital in June 2023, where she built a comprehensive anatomic and transcriptomic taxonomy of adult mouse spinal projecting neurons. Carla's efforts in characterizing SPNs, a major population of neurons infected by insults such as spinal cord injury and amyotrophic lateral sclerosis, are motivated by her longstanding interest in neural injury and recovery. Before starting her MD PhD at Harvard, Carla completed an MSc in regenerative medicine at the University of Galway under a George J. Mitchell Scholarship and a BSc in bioengineering at the University of Pennsylvania. After her MD PhD training, Carla aims to work as a physician scientist towards developing better strategies for neural recovery following injury or disease. Please take it away, Carla.
Hi, everybody. I'm Carla. Thanks for the grand introduction. Let me know if you're seeing the right screen.
Looks great.
Thank you. So today I'll be discussing our efforts in collaboration with the Allen Institute to develop a brain-wide atlas of spinal projecting neurons. As you all know, our brains allow us to execute extremely diverse functions, such as voluntary and involuntary motor functions, as well as they help us to modulate our senses, emotions, and autonomic states. These behaviors involve varying levels of control. Actions like walking require coordination of whole body movements. Playing the piano relies on fine motor control of individual fingers. And states like sprinting and sleeping represent two examples of coordination that is needed between the autonomic and motor systems. A key question in neuroscience, and one that is fundamental to the Zhigong He lab, is how does the brain generate these diverse actions? The answer to this question, at least in part, likely resides within the neurons that connect the brain to the spinal cord. We call these neurons spinal projecting neurons, or SPNs, as I'll refer to them in this paper, in this presentation, and they act as an information highway between the brain and the rest of the body. Decades of research have provided great insights into the relationship between their anatomy and their broad functional organization. So, for example, we know the major descending pathways arise from the cortex, red nucleus, and reticular formation. And functionally, we know these major descending pathways have classical actions. For example, the corticospinal tract in fine motor control and the reticulospinal tract in posture and locomotion. We've also begun to appreciate that their molecular and cellular diversity likely also underlies their diverse functional outputs, but there's many major gaps that we have in understanding their cellular diversity. Primarily, we don't have molecular markers for many of these spinal projecting tracts. Also, we don't know how many cell types there are within each of these tracts. And importantly, we don't know how cell type diversity differs across these anatomically defined tracts. So in an effort to fill in these major gaps, we sought to build a cell type atlas of spinal projecting neurons. To do this, we use something very similar to the previous presentation. We use RetroSeq, in which we retrogradely label SPNs via injection of a retrograde AAV into the spinal cord of adult mice. And then at P56, we dissect out labeled brain regions, and then we sort fluorescently labeled nuclei using FAMs. And then we perform single nucleus RNA sequencing with both 10x and SmartSeq. In parallel, we assessed the anatomical distribution of SPNs across the whole mouse brain using serial 2-photon tomography, or STPT. So segmentation and registration of our labeled nuclei to the Allen Common Coordinate Framework showed that SPNs are widely distributed across virtually the entire mouse brain, but have concentrations primarily in the cortex, hypothalamus, midbrain, pons, medulla, and even the cerebellum, which is all consistent with published literature. After applying standard quality control metrics to our single nucleus RNA sequencing data, we generated about 65,000 high quality profiles, which are shown here in UMAP space. We then employed a multi-level iterative clustering approach, similar to what has been presenting already today, to generate this hierarchically organized transcriptomic taxonomy of SPNs, which is depicted in this dendrogram. I'll dig into this in a couple of slides, but briefly, our taxonomy divides into 3 main divisions, 13 different subclasses, and 76 final SPN types, all across these 8 dissection regions. To validate our taxonomy and precisely map these 76 SPN types to their spatial locations, we leveraged the impressive data set presented by Zizhen Yao earlier today. So as a reminder, the Allen Institute generated 2 single cell level whole brain scale data sets, the first being a single cell RNA sequencing data set of about 4 million cells after QC, and a MERFISH data set of also around 4 million cells. So first, we mapped our SPN taxonomy to the whole brain single cell RNA sequencing data set, and we find good cell type correspondence between our SPN types and those presented in their paper. And second, crucial to our study, we integrated our SPN taxonomy with the whole brain MERFISH data set to spatially localize the SPN cell types, which I'll step through in the next couple of slides. So looking at our dendrogram and MERFISH integration results, we find that this first division corresponds to SPNs arising from the cortex in layer 5, red nucleus, which are rubrospinal neurons, and deep cerebellar nuclei, which are cerebellospinal neurons. We find that they have relatively simple transcriptomes comprising only 7 of the 76 types in our taxonomy. And we also find that they're marked by very discrete, easily definable marker genes, most often just 1 gene, and are all exclusively glutamatergic. Interestingly, from our cervical and lumbar injections, we see that these populations exhibit distinct somatotopic segregations. So these homogeneous excitatory and somatotopic SPNs suggest that they are suitable for point-to-point communication between the brain and the spinal cord. And it's likely more their wiring rather than their identity differences within each tract here that cause their diverse outputs. So an example are corticospinals from the primary motor and somatosensory cortices, which we find to be very transcriptionally similar, but they're known to terminate in different spinal laminae and govern fine movement and somatosensory processing, respectively. Division 2, with 49 types, is our most transcriptionally complex division. They exhibit extremely rich and complex gene expression patterns and contain neurons that are glutamatergic, GABAergic, lysinergic. And interestingly, our MERFISH integration maps this division to the reticular formation, thus identifying them as the reticulospinal neurons. These highly heterogeneous excitatory and inhibitory reticulospinal neurons suggest that their diverse actions might be dictated by diverse cell type identities. So as a toy example, we can have different types of neurons coming from the same region in the reticular formation, might even project to the same spinal cord region, but command different functions. Finally, we find division 3 contains modulatory SPNs that express unique neurotransmitter profiles. They express either slow-acting neurotransmitters like noradrenaline and serotonin, or are marked by abundant neuropeptide expression. So very briefly, this division houses SPNs in the hypothalamus, midbrain editor Westphal nucleus, locus ceruleus, and raphe nuclei. So these modulatory neurons likely act as somewhat of a gain setting system for brain to spinal signals. To put this more concretely with a specific example, early studies in the 1980s showed that neurons from the raphe and LC project to and have a facilitatory effect on the ventral horn, which is the motor part of the spinal cord, and have an inhibitory influence on the dorsal horn. So functionally, the simultaneous action of the dorsal and ventral horn projections should lead to a state of fight or flight, where we have enhanced responsiveness of the motor system accompanied by decreased pain transmission. So taken together, our taxonomy provides the highest resolution view into brain-wide SPNs to date. We find that there's a 3-component transcriptomic organization of SPNs, where division 1 houses corticospinal, rubrospinal, and cerebellospinal neurons, which are suitable for this point-to-point wiring and function for independent movement of the extremities. Division 2 houses our heterogeneous reticulospinal neurons, and division 3 houses our modulatory neurons, which likely are for a built-in gain-setting system. Interestingly, these 3 divisions nicely correlate with a theory of motor systems proposed several decades ago. In the 1980s and 1990s, it was proposed that the somatic motor system has 2 components: a lateral pathway, which likely controls independent movement of the extremities, and a medial pathway, which controls coordinated body movements. And then an additional emotional motor system was proposed, which was thought to mainly contain these modulatory gain-setting neurons. So it seems that our divisions 1, 2, and 3 nicely correlate with this theory proposed several decades ago. So these results are just a taste of some findings from our publication. There's much more in our paper that I didn't have time to discuss today, so I encourage you all to take a read. And lastly, thank you all for listening, and a huge thanks to the numerous people who have pushed this project forward. In particular, thank you to our collaborators at the Allen Institute, Hongkui Zeng, Vasilka Tasik, Kimberly Smith, VK Lee, Zizhen Yao, and Cindy Den Veldhoven. Their help was absolutely crucial for this work. Thank you.
Thank you so much. I'd like to next introduce Hailing Shi. Hailing is a Helen Hay Whitney Foundation postdoc fellow in Dr. Xiao Wang's lab at Broad Institute and MIT Chemistry. She received a BS in chemistry from Peking University in 2014 and a PhD in chemistry from the University of Chicago in 2019. Her research interests are to develop and apply chemical, molecular, and spatial profiling tools to understand the molecular basis of human health and diseases for new therapeutic strategies. Hailing's work has been recognized by awards, including the Trainee Professional Development Award from the Society for Neuroscience in 2022 and the Best Thesis Award at University of Chicago in 2020. Please take it away. Thank you.
Thank you, Yasmin, for this nice introduction. I hope my slides are showing well. And thank you for this opportunity to present our work on spatial atlas of the mouse central nervous system with molecular resolution collected by the platform StarMap. This work is a joint effort between Dr. Xiao Wang's lab at the Broad Institute and Dr. Jia Liu's lab at Harvard. Graduate student Yichun He at Liu Lab and Dr. Yiming Zhou are also significant contributors to this work. Okay. So spatially resolved transcriptomics enables measures of cellular expression in their native context. Our lab is particularly excited about imaging-based spatial transcriptomics methods to capture single-cell resolved gene expression in 3-dimensional space and integrate multimodality measurements of the same sample. And I don't have to emphasize that for the brain, a single cell and a spatially resolved mapping would greatly benefit our understanding of the molecular architecture of brain physiology and pathology, as well as their regional specificity. Besides the molecular characterization of the tissue architecture, the other part of this project relates to the labeling and the manipulation accessibility of other cell types by viral tools, such as recombinant adeno-associated virus, a critical step to probe and modify cell type functions. One of the viral variant, PHP-EB, achieves central nervous system expression by penetrating the blood-brain barrier in mice. So in this work, we also use the spatial transcriptomics-based quantification, aiming to provide a brain region and a cell type result viral tropisms of this variant PHP-EB. The platform we use is an imaging-based spatial transcriptomics platform called StarMap, which my PI developed. This method converts a tissue sample and its RNA into a sequencing hydrogel, revealing single-cell gene expression in its native environment. It combines in situ RNA hybridization using a pair of probes to allow target-specific amplification, tissue hydrogel chemistry to covalently anchor the amplified products into the hydrogel, and in situ sequencing with confocal microscopy to decode RNA identity. With 6 rounds of imaging, StarMap can map over 1,000 different genes in the same sample with high sensitivity and accuracy. So with this platform, we mapped 1022 genes expression in both coronal and sagittal slices from adult mouse brain. Target RNA are mapped at molecular resolution, and after segmentation into single cells using an algorithm called cluster map we developed in the lab, we can get cell by gene expression matrix with the cell spatial coordinates. Just to showcase how the star map raw data look like, on the left is the full hemisphere of mouse brain slices in first round of imaging, and zoom in to one of imaging tile in the sixth round of imaging, and further zoom into a few individual cells. So in those images, individual dots represent a amplified product generated from a single RNA molecule, and they appear as in different color across the 6 rounds, which translate into their gene identity. So in total, we collected a data set of over 1 million cells with 1,022 genes mapped with spatial coordinates. So with this spatially resolved single cell gene expression data, we first performed a 2-layer cell typing by integrating with a paired single cell RNA sequencing data of the mouse brain and label transfer for main cell types, followed by further latent clustering within each main type to get subclusters. So in total, we defined 230 subtypes and then plotted them back into their physical coordinates, get the spatial cell type maps. The molecular maps clearly delineate the brain structures, highlighting the molecular foundation underlying brain anatomy. Plotting the spatial maps of individual subtypes allow visual evaluation of their pattern. And here are examples of the excitatory and inhibitory neurons in the telecephalon, showing different degrees of regionalization. Besides the cortex, we also provide spatial maps for neural cell types in the thalamus, hypothalamus, in the brain and other brain regions. I want to highlight this one example, a histaminergic neuronal cell type. Although it only accounts for 0.01% of the cell population in the atlas, we were able to still faithfully identify them with the expected marker genes and expected spatial location as the ventral media hypothalamus, highlighting the power of star map for spatial molecular cell typing. Next, to further utilize the spatial information in the dataset, we want to build a molecular definition for tissue regions. And such data-driven identification of tissue regions can provide a systematic and unbiased definition for the tissue regions. We chose to define molecular tissue region by directly using spatial niche gene expression profiles to fully utilize the high-dimensional data we have in this 1022 gene spatial mapping. Briefly, a spatial niche gene expression vector of a single cell is made by concatenating the single cell expression of the center cell self with its k-nearest neighbors in the physical space. The resulting matrix are concatenated and integrated for clustering to generate molecular defined tissue regions. We also registered the coronal sagittal slices into the established Allen mouse brain common coordinate framework to get an anatomical definition, allowing the comparison of differences and similarities between the molecular definition and the anatomical definition of tissue regions. Here is a heat map showing the distribution of individual molecular cell types across the molecular tissue regions, and consistent with many accompanying papers in this package, we find majority of the neuronal cell types have high regionalization concentrated in certain brain region. By comparing the molecular definition and the anatomical definition of tissue regions, we find the cases where the molecular definition can provide refinement to the anatomical definition. One of the example is from the retrospinal cortex. In the anterior part of the brain, we identify 3 molecular tissue regions corresponding to anatomical retrospinal cortex with clear marker cell type and marker genes. However, posteriorly, the same molecular tissue region correspond to a different region, the subcilium, pre-subcilium, and post-subcilium. And in between, both the anatomical retrospinal cortex and the subcilium are covered. So this observation may give insights into a recent study showing differential functions in the anterior and the posterior RSP. So we propose a molecular tissue region could facilitate delineating the retrospinal cortex and its subregions on top of the cellular and tissue morphology. We also observed a case where the joint single cell and spatial definition of cell types can resolve cell heterogeneity better than the single cell gene expression alone. This example comes from dentate gyrus granule cells, which largely form a homogeneous cluster in the single cell gene expression space. However, they fall into two distinct molecular tissue region clusters corresponding to the dorsal and ventral part of the dentate gyrus in the hypothalamus with enriched gene expression. So this unique tissue region segmentation through spatial niche gene expression may provide insights into the functional divergence around the dorsal ventral axis of the hippocampus. Switching gears to the viral tropsin characterization, in collaboration with Deverman's lab at Broad, we labeled the PHP EBAV genome with a barcode and map the viral expression together with cellular expression on the same sample. Spatial AAV expression heat map shows a prevalent expression in the brainstem, especially the thalamus compared to the cerebral cortex. We also quantified the PHP EB transduction in individual main cell types and subtypes. Here's an example for showing the importance of deep cell typing in this type of tropsin profiling. We see subtypes of astrocytes actually have a varying degree of viral transfection. So in summary, this work demonstrates the incredible scalability of StarMap platform and the accompanying analysis pipelines to build a spatial cell atlas of mouse brain, comprising over 1 million cells and over 1,000 genes mapped at submicron spatial resolution in all 3 dimensions. With spatially resolved single cell expression, we achieve spatial cell typing, identify molecular tissue regions to refine tissue boundaries. We also expand these gene lists to predict transcriptome-wide spatial gene expression in single cells. In addition, we categorize the tropsins of common viral tools across cell types and tissue region. Finally, as a part of a community, we value open access. We have a data portal for everyone to explore their favorite genes or cell types. All the data and the code have been made available online for the community to get a data annotation and share this roadmap of spatial single cell analysis. With that, I will end with thanks to everyone who make this work possible, and thank you for your attention.
Thank you so much, Hailing. Next, I'd like to introduce Ethan Armand. Ethan is a PhD student in the Ren Lab at UCSD. Ethan's research emphasizes using epigenome measurements of the mammalian brain to decipher the gene regulatory code and identify functional non-coding elements in the genome. Please take it away.
Thanks, Yasmin, for the introduction. Real quick, I want to sort of highlight this work was done with a lot of collaborators, but particularly Nathan Zemke, the other lead author of this work, was obviously super hugely influential to all parts of this. I hope that, or I'm sure my colleagues have sort of highlighted that cis regulatory elements are really important to defining cell types and cell type specific gene expression. But I want to highlight that it's also a key driver in evolution across species. You can see, starting from a cis regulatory sequence, you might have some sequence changes which result in different activity of cis regulatory elements. And ultimately, while the proteins themselves are rarely changing across species, the level of expression of these various genes and proteins is what contributes a lot to this sort of evolutionary diversity. And so to understand the evolution of these elements, we looked at the primary motor cortex of 4 species spanning around 90 million years. And we were able to generate integrated cell type maps of around 20 cell types or 21 cell types across all these species. And for each of these cell types, we're able to measure 4 modalities. So we have 3D genome interaction, obviously gene expression, chromatin accessibility, which is sort of helping us define these regulatory elements, as well as cytosine DNA methylation, which again helps us define these regulatory elements and map the functional ones to the genes that they target using the 3D genome. And we're able to profile this in every cell type in every species and generate these really amazing comprehensive maps of the gene regulatory landscape of each of these cell types. Given we have this data, we sort of developed a methodology to identify what's conserved and what's divergent. Conserved programs are often like core important functional programs, and of course divergent programs allow us to look into the specifics of evolution. So we sort of measured, if you know the expression of a gene in one cell type in one species, how well can you use that information to predict that in a different species? We measured this using a regression to identify divergence. We compared using traditional differential gene expression. And this allows us for each gene and each regulatory element to sort of assign them a level of conservation divergence, identify the highly conserved elements, identify the highly divergent elements, which are specific to one cell type or one species. Taking a step back and sort of using this knowledge to look at transcription factors, which have been highlighted over and over again today, and the way they contribute to evolution, we find that as the transcription factors of a cell type have more divergent expression, it's reflected in an epigenome that's becoming more divergent in a very concordant manner. Looking at how these same transcription factors influence the conservation of the elements they're active in, we find there are a lot of transcription factors that are really representative of conserved sequences, while other transcription factors like OLIG2, FOXP2, for example, might be more represented in divergent cis-regulatory elements. Along with the hypothesis that conserved elements tend to have core important functions, we find that conserved distal elements are much more likely to be algorithmically predicted as being an active enhancer and assigned to a particular gene. And to sort of validate this, we look at H3K27 acetylation, a well-profiled enhancer mark, and find the same trend. And sort of to further validate the utility of these comparisons, we find that when looking at fine map SNPs that fall into cis-regulatory elements, the cis-regulatory elements they fall into are significantly more conserved than a background set of random elements. But of course, I sort of started this talk promising that we would be able to link element change to actual changes in transcription. And of course, we did that. To start off, if you look at a enhancer with a human-specific regulatory activity, and you find the gene that it's predicted to regulate, on average, those genes are significantly more divergent across species. And we can actually find all of our significantly human upregulated enhancers and the significantly upregulated human genes that they target, and looking at the sort of p-value of that comparison, whether it's how upregulated the enhancer is or how upregulated the gene is, we find a really striking correspondence. And some of these genes are, famous language evolution genes like FOXP2, for example. And so we can actually go into this FOXP2 locus, which is expressed specifically in human microglia, and identify a likely causal enhancer, which has increased chromatin accessibility in humans compared to every other species, and increased levels of acetylation, sort of validating it as an enhancer that's likely responsible for taking this generally excitatory neuron active gene in the cortex and turning it on in microglia. And we also find RIR3, a calcium release regulating gene that in other species is again sort of restricted to neurons, has high levels of expression in human astrocytes, given a few putative regulating enhancers, which are likely turning it on. So given all this data, we wanted to understand how well do the gene regulatory landscapes we identify in one species, given they're driven by transcription factors, which are core to cell identity, can we, from sequence alone, understand the epigenome of a species? And using this, we used a deep neural network approach. And for a first sort of question is, are these the regulatory landscapes of these species similar enough that we can use them to augment our training? And we find that by including either more modalities of data or more species in our training, we get a very significant uplift in accuracy when we're trying to predict the activity of a region we've never seen before. And this is really well represented when you look at the track. Everything with a sort of yellow dot is the prediction our model made. Everything with a purple dot is the true measurement. And for this, for the Huntington gene locus, we see a pretty remarkable correspondence between our predictions and the true values. And as sort of a final validation of this, we wanted to see, do we even need to include a species to be able to predict its epigenome in these cell types? And we find that overall, even in our worst case scenario, where you have a model that is representative of the most evolutionarily distant species, across our species, we get accuracies that are almost equivalent to what we would get from training on the original species just itself, sort of showing a remarkable generalization here. To conclude, I just want to thank everyone involved in this work. Highlight again Nathan Zemke, Soyun, and all the other involved labs for their help. This was a huge effort, and thanks everyone for your time.
Thank you so much. I'd like to next introduce Karthik Shekhar. Karthik is an assistant professor in the Department of Chemical and Biomolecular Engineering at the University of California in Berkeley. His laboratory is cross-affiliated with neuroscience, vision science, and the Lawrence Berkeley Laboratory. His interests are at the interface of neuroscience, genomics, and applied mathematics, and his group uses both experimental and computational approaches to understand how diverse types of neurons in the brain develop and evolve and become selectively vulnerable during diseases. He has received the NIH Pathway to Independence Award, the Hellman Fellowship, and the McKnight Fellowship in Neuroscience. He also recently received the Donald E. Noyce Prize for Excellence in Undergraduate Teaching at UC Berkeley. Please take it away. Thank you.
So thank you very much for this opportunity, and I thank all of you that are still listening to this these presentations. So this talk is about the retina, and the retina, although it's located in the eye.
Karthik, could you swap the screen, please?
Oh, okay. There we go. Is it better?
Yes, perfect.
Thank you.
Right. So the retina, although it's in the eye, it's very much a part of the brain, and it's the tissue that all of us use for seeing the world. So the basic architecture of the retina is extremely well known. The retina has a laminar organization comprising 6 major classes of cells, beginning with the photoreceptors in the outer layer and various classes of interneurons in the middle, and all the way to retinal ganglion cells, which are the projection neurons. And it's also been known from the time of Cajal that each of these classes can be subdivided into multiple cell types, and there have been a long history of works using morphology, function, and molecules, and more recently transcriptomics that have classified these cell types. What's also interesting about the retina is that its basic blueprint is really evolutionary ancient. We can't say that about the cerebral cortex. So the retinal blueprint comprising the 6 major cell classes are actually shared between primates all the way back to lampreys and the early jawed fish. And so the question is, how then do these different species have wildly different visual behaviors? And so there must be some adaptation and evolution at the level of retinal circuits. And so here we decided to ask how conserved are these retinal cell types across species, and hopefully this will be a foundation to assess conservation at the level of circuits. I just want to point out some interesting things that are known. So for example, it's well known that in primates, we have these interesting pathways. These are called midget pathways that allow us to see at very, very high resolution. And this is dictated by these midget ganglion cells. And where if we go in the periphery of the retina, on the other hand, you don't see these one-to-one connections. The connections become more many-to-one, and that's why we have very low resolution peripheral vision. And these midget pathways have so far not been identified in non-primates. So the question is, how do these evolve and what is their origin? So we decided to answer this question by collecting single-cell transcriptomic data across 17 species and integrating them. And our species include, primates, several rodents, some ungulates, and we also have some non-mammals like zebrafish and lamprey and chick. And this project was led by Josh Hahn in my lab and Abuzar Monawar Feshani, who was a postdoc in Josh Sains' lab, and all of this is an equal collaboration. And so what do we see? So the first thing is that I mentioned about these 6 classes. Again, unsurprising, but the transcriptomic signatures of these 6 classes are very well conserved across evolution. And what we see is that in general, the class identity dominates over species identity. And we can also see this in the conservation of several transcription factors across these different classes among all the species that we analyzed. And so what this suggests is that the conservation of these transcription factors also suggests that the developmental mechanism, which have been extensively analyzed in mice, may also be conserved across the entire evolution. So these mechanisms are truly ancient. But then what's happening at the level of cell types? And this is what we were really interested in, and this was the subject of our paper. And so here I'm again showing the retina, and we dug deep into the cell types within 2 classes, one being the bipolar cells and the other being the ganglion cells. So the bipolar cells are key excitatory interneurons, and the ganglion cells are the projection neurons. And the reason we chose to focus on these classes is because we know hell of a lot, both in terms of morphology, physiology, and also transcriptomics across both of these classes, especially in mice, and to some lesser extent in primates. But of course, we wanted to understand the evolutionary relationship across a much larger set of species. I want to also just point out that in an earlier analysis where we compared mice and macaques, one species of primates, what we also find is that the cell type variability actually increases from the outer retina to the inner retina, again, suggesting the hypothesis that what nature is really doing is diversifying the cell types within ganglion cell layer, because this is what is being projected from the eye to the brain. Okay, so conceptually, just as we know the concept of orthologs, which are 2 genes in 2 species that descended from the same ancestral gene, we want to identify orthotypes, which are 2 cell types in 2 different species that presumably descended from a common ancestor. And of course, here what we do is we integrate these different gene expression data sets, and here we are using one-to-one orthologs. And our hope is that by clustering these data in a single latent space, these clusters may represent these orthotypes, which are groups of cell types across different species that have a common gene expression signature, and by implication, a common evolutionary origin. So what do we see? And so when we analyze the data without any integration, unsurprisingly, the cells, bipolar cells displayed here, clustered by species. But the application of integration methods sort of beautifully collapses all these different cell types into these common clusters. And one can then cluster this latent space and identify these orthotypes. What's also interesting is that if you now look at how these orthotypes are represented across different species, and this is a large confusion matrix where the rows represent these orthotypes and each block represents a species and the columns within a block represent those species-specific cell types. What you can clearly see is that every orthotype corresponds to one cell type, typically in every species. And the inescapable conclusion is that all these cell types were likely present in the common mammalian ancestor, and that's why we inherited them. Our ability to match these correspondences is slightly lower in the non-mammals, and that's partly because of the paucity of data and also likely an improvement of methods that are needed, and we are working on that. So now what happens in ganglion cells, which are more variable? To our surprise, we also found that ganglion cells were extremely well conserved. And I want to cut to the chase and talk about these midgets and parasols, which are the dominant types in primates and especially humans. So each of these cells is what's called a paramorphic pair. There's an on type that responds to illumination and an off type that responds to darkness. And these cell types, midgets and parasols, almost make up about 95% of the retina. So they're very important for our vision, and they're also clinically important. And as I mentioned, no equivalents of these types have been found in non-primates, and they're currently thought to be a primate innovation. But to our surprise, we found clear orthotypes that contained these midgets, each of these sort of midgets, on midgets, off parasols, and off parasols. And these orthotypes contains cell types from several other species, okay, including mice, suggesting that perhaps these cell types have clear orthologs in other species, and therefore they must have been present in some form in the vertebrate or the mammalian ancestor. And what's interesting is that although this prediction is purely based on transcriptomic similarity, we find matches in several other modalities. So for example, there is a great match at the level of morphology and dendritic lamination. They also match based on their physiology and the kinetics of light responses, although none of this information was provided explicitly. They also match in terms of response type. And what's interesting is that the major presynaptic bipolar neurons to each of these types in mice and humans also happen to be part of the same bipolar orthotype. So that was quite exciting. So outside of these matches, there's a major difference in that the relative frequencies of midgets and parasols is almost 70 to 90% in primates, but in rodents, these orthologs make up a minority of cell types. And what we can see is that what's really happening from left to right is that there's also an increasing role of cortex in visual processing. So what we think is really happening is that the corresponding to the major evolutionary shift between mice and primates from the superior colliculus being the major retinal-recipient region to the DLGN, what we see is that these so-called mouse orthologs of midgets, despite being a very small proportion of mouse retinal cells, they disproportionately project to the DLGN, kind of suggesting that it's this path, as this pathway expanded from rodents to humans, the corresponding cell types also expanded in number in the retina. So what I want to sort of leave you with is the main takeaway, which is actually an evolutionary hypothesis, which is that as the cortex expanded in primates, the cell types present in the vertebrate ancestor, they decreased in size and increased in number to become the midget cells that provide the cortex with a high acuity, minimally processed image of the visual world. So we think that's what's happening. And with that, I'd like to thank my group and all the funding agencies. I want to acknowledge that this was an equal collaboration with George Sainz' group, and our data and code and notebooks are all publicly available. Thank you very much.
Thank you so much. I'd like to invite all the speakers to now turn on their video and join us for a Q&A session. I had a lot of great questions coming through the Q&A, so thank you to all in attendance for great questions. And the speakers have tried to answer most of your questions, including the ones that Jimena added, which were submitted in advance through registration. Apologies. So I wanted to pose a question to the group. And this is one that's already been posed, but I think it's good for everyone to hear. Is there a unified integrated map of all those transcriptional data sets presented in these different papers? And if not, do the taxonomies and clusters match between the data sets? Like, could you follow the same molecular cell type across atlases? Thank you for the question.
Yeah, maybe Zizhen, do you want to take the lead in answering the question? Yeah.
Yeah, I think we have because of the timeline for this publication, we couldn't do it in like very thoroughly when the paper published. But now, after publication, we have done more, careful integration between at least our taxonomy versus the single nuclear RNA-seq atlas from Broad. So our initial assessment shows there was a tremendous, agreement between these two atlases, considering one is primarily driven by single cell and the other is single nucleus, and then we have slightly different sampling strategies, and then there were more than 5,000 clusters in both atlases. The agreement is tremendous. But having said that, there were still details that need to be resolved before we propose the final integrated taxonomy, because in either case, there were different decisions made how to cut up the gradient, for example, and there were some parts of the brain that, due to the extreme heterogeneity, and there was more challenge in some part of the brain than others to completely resolve it. And I think maybe that reflects also some additional sampling that is needed to completely resolve those types. So, in summary, yes, I think we can provide preliminary correspondence between the cell types, have a translation table between the cell types between two taxonomy, but it will take more time to completely integrate them into one taxonomy. Yeah.
Thank you. Anyone else wants to answer? I think we can move on to the next question. And this is kind of along the same lines. Are there discrepancies in the scRNA-seq data sets from different groups, like between Alan Zeng and Brod-Macosko group and more?
Oh, can you elaborate the question again?
Sure. This is a question from the Q&A. The question is if there's a notable discrepancy between scRNA-seq data sets from different groups. And I think Jonah answered this one in the Q&A.
Jonah, maybe you can comment a little bit more.
So there is single cell versus single nuclei. I guess most of what I had to say is in line with Zizhen. I think there is interesting sampling strategy differences, and Zizhen said some technologies can be more or less compatible with dissociation across different tissues. And so I think we've been amazed by the detail of both data sets. So I think they're both very positive, and they look at the problem slightly differently.
Sounds great. Thank you. This is a question that came in advance, and the question is kind of a broad one, which is how do we establish a correlation between different cell types and the respective functions? I think, Hongkui, you answered this one.
Yeah, I think this also ties with the previous questions, the reconciliation between single cell and a single nucleus. I think it goes beyond that, right? In the end, we want to define cell types and also better understand cell types by incorporating other properties of the cells as well. Their connectivity, as shown by several papers here, and also evolutionary comparisons, which I actually didn't emphasize in my introduction, but it's a very important way to understand the relationship between different types and also the different methods, as Ethan and Karthik's talks illustrated very well. And ultimately, we want to align cell types with their function and to see if different cell types have specific functions or not. So this is going to be a long way to go. The starting point that we propose is to use the marker genes and epigenomic landmarks that we already identified as a way to mark different cell types for functional studies. And then furthermore, creating genetic tools that will allow us to label and manipulate specific cell types in vivo context and to study their function. So the genetic tools would be created by the combination of transcriptomics data and epigenomics data, identifying cell type specific enhancers as shown beautifully in Hanqing and Songpeng's presentations as well as Jingtian's, and test them. And if they're validated tools, then it will facilitate a lot of the functional studies and understanding the roles of the cell types in circuit and in behavior.
Thank you so much. I think we have to end it there, unfortunately. Thank you all for your great talks. I think we all learned quite a bit today. And thank you, audience, for participating in this workshop, for this webinar, and for your great questions. We weren't able to answer all of them, but we hope you learned something useful today. Thanks again for your participation. Please complete the feedback survey that will pop up as you leave, and the links also in the chat. We'll be posting the recording in about within 2 weeks on the BICAN YouTube channel. Thank you and have a good day.
Thanks, everyone. Thanks to all the speakers for the fantastic talks.